Hello. So my point today is to speak about the fact that each of you has a unique cortical folding pattern. And I will describe a program that aims at leveraging that fact to get some clue about some specificities of your architectonic organization. And maybe more importantly, we would like to define what is abnormal folding pattern, because it's of interest, for instance, to drive epilepsy surgery. And currently, there's no clear way to define what is abnormal. Okay, so the link with eBrains is very clear. It's that for people who know eBrains, there's several template space into eBrains where you can upload some of your data sets. And we had to develop a way to go from one of these spaces to another one. And we decided to force the fact that the main sulci are aligned between these spaces. And this could be discussed. This is clearly something to be done in primary areas, but elsewhere it's clearly not so necessarily the best way to deal with that. And what we have in mind there is that when you're dealing with single brands, like the big brand, it's a very interesting issue to ask. It could be that, for instance, to map architectonic area in a specific brand, rather than taking the big brands or the Yiddish brand, the best would be to choose among the brands that have been used to do a Yiddish brand, the one which has the closest folding pattern to this specific brand. So, just to clarify for the naive people across you why it's a complex issue to link brands using folding, I will just show you two specific brands chosen randomly for you to realize how complicated business it is. So these two brands are very, very different. It's the white matter of these brands. If you're familiar with brands, rapidly you discover that there's something stable between these two brands, which is the central sulcus, which is hosting primary sensory motor areas. So, of course, because of the strong link with architectonic, these sulci should be matched across people. If we move to superior temporal sulcus, the situation is more complex. You realize that there are some interruptions of this sulcus. It's not the same in the two brands, so there's open issues there. How do we deal with this interruption when we try to align these brands? And if you go to the frontal lobe, it's a nightmare. There are colors there, which are colors maybe proposed by me as a link with the standard nomenclature, but I probably have done some mistakes. And driving the alignment using these colors is problematic. The community is just hiding the problem under the carpet by doing that without any labeling, without any identification, but the problem remains the same. What's the link between these brands? And of course, for a lot of people, there's no link. This is just folds that are used by nature. It's a trick to put more cortex in a skull, but you should not pay attention to the specific folding pattern that you discover in a specific brand. But the situation is probably different. For instance, with this very interesting work that has been done on a big brand by the McGill and the Yiddish team, they have clarified that not only there's difference in thickness between the bottom of the sulci and the giral crown, but there's even a difference in the relative size of the different layers, which means that at least there's an impact of folding on the organization of the microstructure. But maybe it's just mechanics, maybe it's not related to architecture. And if you look at this work, which is incredible work by the physicists, which are interested in folding these days, with a very simple equation that is just modeling the fact that the gray matter is growing faster than the white matter, especially in the tangential direction, you can create these simulations which are really looking like a real brain. It's the geometry of a real brain without any architectony. This is really pushing to the direction that this is just random folding. We should not pay attention to it. Even more people like me who are trying to put some link with architectony, it's not at all the situation of proteins where the shape, the form is doing the function. It's not like that with folding pattern, of course. And the physicists have pushed the story further away, triggering their simulation from the shape of a fetus that comes from MRI. They get some simulations that are looking more and more like a real brains. The directions of the folds are quite similar to the direction of the fold in the real brains, but the folds are not at the right place. So what is missing in this simulation to get something looking like a real human brain is biology. And there was a lot of changes in biology during the last 15 years. It has been discovered that species with folds have a different neurogenesis compared to species without folds. Mars and human brains have a different trajectories of migration. What's happening is that in the ventricular zone, you have the glial cells that are building the ladder towards the cortical plate. But in gyroscephalic species, there's another zone which is called the outer subventricular zone, where there's another kind of glial cells that is adding other layers and is creating some divergence leading to the folding. And the interesting thing is that the gene expressions leading to the proliferation of these different kind of cells is heterogeneous. If you look at the ferret brain, there's a section with a tracer of these proliferators, and you see that there's much more proliferators under the future gyri, what may be called the proto-gyri. So here, the idea is that there's something a bit similar to the idea of Reiki on the protomap of architectony. There's a protomap of folding that is driving, in fact, the whole process, and which is looking like a mosaic of places that are future folds and places that are future chiral crowns. So my point there is that maybe differences in the genetics of that, because of what's happening there is that the expression of genes is not the same in these different kind of places. Maybe differences across people leads to different folding patterns, so you can guess what happened during the early neurogenesis. So what I have in mind there is that if you look at a brain, you see some patterns looking like letters or ideograms, and you find, for instance, the same pattern, the same letter in a group of subjects, and another letter in another group of subjects in the same place, and it's corresponding to slightly different neurogenesis, leading to maybe slightly different architectonic organization. So just to clarify what I'm calling folding pattern, and it's probably the reason why I'm doing this talk here in Croatia, which has a very strong reputation for working on the development of the cortex. It's the idea that this is something that appears in utero, before birth, and will never change. But folding is changing during our whole life. During the first years, of course, the folds get deeper, their length increase because the brain is getting bigger. During adolescence, in fact, the process flips backward because of the development of myelin that is taking a lot of space. So white matter gets inflated, so the cortex is unfolding. And later on, because we are losing neurons, of course, the deflation is going further away and sometimes accelerating in case of neurodegenerative disease. But the patterns I'm talking about, they are stable during the whole life after birth. So if we go back to this superior temporal sulcus, these two brains, the vision we have there is that for the folding dynamics, there's two attractors. Some of the brains fall in the attractor of the left pattern, and other ones in the attractor of the right pattern. And this is just related to some elements of the genetics expression, or maybe the size of the brain, things like that. And just to clarify a bit further this vision, I will use another model of folding, which is less realistic than the ones that comes from physics. It's something that comes from the idea of Turing, of generating patterns in the real life using the interaction of two morphogenes. And when you use this system to mimic, in fact, folding, you get something interesting, which is that according to the parameter domain, and there's only two parameters in this equation, which is why it's interesting as a model, you can get a standard folding pattern or a polymicrogyri or a lysencephalic brain. So the idea there is that you have a phase diagram with two parameters, and according to where you are in this diagram, you will get a different folding pattern. And if we focus in the ecological zone where the folding is looking like the folding of normal brains, you can imagine that there are some smaller attractors in the spirit of what I was mentioning for the superior temporal sulcus. And each topological pattern of a specific sulcus is corresponding to one of these attractors. And these attractors are acting very early during the dynamics of folding in utero, because the fork occurs because of the development of architectony. And these attractors that are described in these slides are probably completely standard. But you may imagine that there are some attractors further away from the ecological domains that may correspond to abnormal gyration, maybe the signature of some events occurring during development and creating some weakness in the brain, leading, for instance, to epilepsy, because, for instance, of a problem of the migration of some neurons. We can go a bit further with this modeling. In fact, one of my friends, a neurosurgeon who has been working on epilepsy surgery for 30 years, has suggested that a way to organize all this variability would be to change scale, not playing at the scale of the standard anatomical nomenclature, superior-temporal sulcus, central sulcus, but at a smaller one, which is the scale of what he calls sulcal roots. Imagine that each of the sulcus of nomenclatures is split into pieces. These pieces are the sulcal roots, and they are the pieces that cannot be split in any brain. And of course, some of them are related to the folding dynamics. It's the first places where you see some folds in some of the brains, but it could be more complicated than that. Some of these roots are just the deepest places in the adult cortex. And if you associate some letters to each of these roots, you realize that the standard nomenclatures correspond to some words, and more unusual patterns correspond to other words. If you look at the brain on the left, this one is a very standard long superior temporal sulcus where all these roots are connected. And the ones in the middle, the ones I was showing before, correspond to different connectivity between the roots. So it's a way to organize, in fact, this zoo of patterns to get a clearer idea of what's happening. At the scale of the whole brain, it's in terms of, there's a combinatorial explosion, which is linked to something difficult to deal with, especially if you want to have a kind of understanding of what is normal or abnormal. There's really a very complex probability function. But in fact, it's not that random because there's a kind of funnel behind. If you look at this average brain coming from McGill, the ICBM 152, this is probably the most standard organization of the folding and the highest probability pattern. But at the global brain scale, it's still very, very complex. But if you do a transition towards a regional organization of these patterns, you just have a few patterns to deal with. And this story can go even further. There are some embryological reasons that lead to a specific organization of these sulcal roots into a grid with two directions, like meridian and parallel on Earth. And in most of the places in the brain, there's a direction of this grid that is leading to the standard nomenclature. For instance, in the central sulcus area, you get central sulcus, precentral sulcus, postcentral sulcus. They are all parallel because these roots connect in what we call the meridian direction. In other places, for instance, in the frontal lobe, it's the other direction. So probably in the general population, if you look regionally, you get a few of these patterns according to whether you get connections in the meridian or in the parallel direction. So for instance, in this cartoon example here, you have three attractors. But if something happened during development, disturbing, in fact, the migration on all this stuff, you could get another attractor push forward. Here, it's just a very simple example where you have agenesis of corpus callosum, and it's well known that it's completely changing the organization of folding in the cingulum area. In fact, rather than having a long fold in what we call the parallel direction, you have a lot of small folds in the orthogonal direction. And this is, with our vision, another attractor, a rare attractor that is corresponding to abnormal folding. So with this vision, we've been trying for decades now to build what we would call a dictionary of all the patterns that you find in the general population, and maybe patterns that are some sign of abnormal events. And it's been really, really difficult, at least for the human brain. With all these very incredible visualization platform that we propose to the people who are interested in this topic to browse the brains. From time to time, we have a hit. For instance, here, what has been discovered from my colleagues in Sainte-Anne Hospital in Paris working on sensory motor epilepsy is that in this specific patient, there's a kind of ironic discovery that half of the people have this pattern that is looking like the power button of computers. And this pattern is very rare in the general population. But browsing a lot of brains, it's very rare that we do this kind of discovery. I think it's beyond the cognitive capacities of humans. It's too difficult to deal with. So we rely more and more on machine learning and artificial intelligence for that. I will just show you two simple results for you to understand what can happen. For instance, even with the central sulcus, which is probably the most stable place in terms of folding, if you use a machine learning technique to discover what we call a manifold, which is a space with low dimensionality that is describing the variability across the population, you discover that central sulci can have either one knob or 2 knobs, or even more knobs than that. And if you use, in fact, this split of the population to do some functional mapping, you realize that the upper knob is really associated to the hand sensory motor areas, but also that the development or not of the lower knob is changing the localization of a reading area associated to language system. So just to give you a kind of illustration of what we can do if we use folding to predict where is architectony, or maybe sometimes why architectony is not standard. In the context of pathology, with the same very simple sulcus, we played a game of trying to discover the same kind of organization with a population of people missing one hand, either because of genetics, they are born with only one hand, or because of amputation. And if you ask machine learning technique to give you a very simple one-dimensional space describing the variability of this population, you discover, in fact, that the people born without a hand have a very flat central sulcus compared to the general population, while amputees do not change. Once again, it's pushing forward this idea that folding pattern is stable during life, even with events like amputation. So it's showing that if you look at folding pattern, you can discover in the people missing one hand that something happened that was just preventing the correct development of the sensory motor area. But dealing with that with the standard nomenclatures is a problem, because when you have weird brains, it's very difficult to match the standard nomenclature with them, and it's disturbing the technique I was just mentioning. It's a bit like if we were trying to study the shape of hands with a model of the standard hands and hands with a variable number of fingers. It would be really, really problematic. So now for a few years, we switched to what is called the non-supervised path of artificial intelligence. So we are just forgetting about the path and trying to reorganize this folding pattern automatically using non-supervised technique. And you will not be surprised to understand that we do that with deep learning. And what we are trying to do is, rather than doing a map of all the patterns that you can find in general population, which is really ill-posed in terms of mathematics, we are trying to look for patterns that allow to predict something about the brains. So typically, if you take, for instance, 1,000 brains of the HCP dataset where you have a mapping of function of diffusion, we are trying to find a way to decompose the folding pattern of this brain that can predict their organization in terms of function or in terms of diffusion. So just to conclude, in fact, I will do a parallel with something that has been very, very advertised during the last years because it's just an incredible event for science. It's AlphaFold. With AlphaFold, with a few 100,000 proteins with known geometry, with known folding, it's possible now to predict the folding of any proteins. So I think our situation with the folding pattern of the brain is a bit similar, because there's a few 100,000 brains that we get from research, biobank, and all the cohorts that we all manipulate. And on these brains, we have a lot of annotations in terms of functional organization, connectivity, or pathology. So it's a kind of Rosetta Stones to try to learn what information in the folding pattern could be used to predict this annotation. And it's the game we are trying to do. We are developing, for the people who know deep learning, some latent space were organized according to this prediction power, and I hope that we'll finally build a dictionary that will be useful. And I will stop there.